Hi, welcome to this lecture. Now, if you have to little bit let us understand what we have learned, and then we will start the understanding about how to fabricate the neural implants. Okay. So, we have learned how the substrate looks like, which is silicon. We have learned what are the substrates, like it can be silicon, can be glass, can be polymer. We have seen physical epideposition techniques. We have seen or understood a bit on chemical vapor deposition techniques. We have seen photolithography process, and we have taken few examples of how to use photolithography to pattern certain materials. We have also looked into etching techniques, whether it is wet etching or dry etching, and within etching techniques also we discussed about RI, reactive etching, plasma based etchant, and deep reactive etching. Now, bulk micromachining and surface micromachining we will take in the next class, because there we will be kind of understanding how those processes can be used when it is a silicon based process technique to fabricate cantilever or to fabricate a diaphragm for certain applications such as gas sensor. But in this class, now what I want to teach you is the fabrication of microelectrode array. Okay. So, now let us first understand why we want to learn this microelectrode array and what exactly this array is. So, let us take an example of epilepsy. Okay. Epilepsy is also known as fit or seizures. Generally, this fit or seizures, they are treated by using, or the medication is called anti-epileptic drugs. But there is epilepsy which is called intractable epilepsy, in which the drugs are not effective. So, to cure that kind of epilepsy, a surgeon has to open the skull, which is craniotomy, reach the area which is causing this seizures or epileptic episodes, and resect that area using surgery. Okay. So, the electrodes that are implanted, because we need to understand, right, which area within the brain is causing what kind of electrical activity and where exactly the seizures are occurring. So, the electrodes which are platinum iridium electrodes, these arrays are there and is implanted onto the brain. Okay. So, this is our skull, this is our head, and you open the head, you will find a skull. When you open the skull, there is a dura. When you open the thin layer of dura, there is a brain in CSF, cerebrospinal fluid. Okay. That part we have Dr. Sabri Girishan, who will be teaching you in detail about the brain and brain related activities, because he is the one who actually implants the device, and he is a functional neurosurgeon, so also implants DBS electrode, which is deep brain stimulation electrode for Parkinson. So, let us understand those details about how it is done through neurosurgeon. I will be talking more about if this is a process, how to implant or how to fabricate this implant and why we need to fabricate this implant. So, the current implant, as I told you, is made up of platinum iridium electrodes, and the electrode size is quite big, and also the resolution is poor because they are not too close, it is not high density. So, we had two ideas. One, can we fabricate a high density electrodes using microfabrication technique? for acquiring the ECOG signal, right. So, what is ECOG? Now, you understand the signals that we acquire from the scalp, right, from the head, they are called EEG signal or electroencephalogram. ECOG stands for electrocorticography. So, when you acquire the signal from the brain using this electrode, we call this as a ECOG electrodes, easy. But we want to have high density electrodes. Why? Because otherwise we need to import this ECOG electrode array, and a customized version is extremely costly. So, how to bring down cost is one of the facets, but the main thing that we are right now focusing on, and I will be teaching you, is how to fabricate this high density electrode microelectrode array. So, one, we have clear idea about why it is used, because we can finally use it in humans. So, that if the resolution is better, the area which is causing these episodes, we can acquire it or we can know it precisely. Second, if we have this microelectrode array and we want to test the efficacy of a drug, which is your anti-epileptic drug, whether the drug is effective or not, right, then we can use or probably use this array to understand the efficacy of the anti-epileptic drug. So, with that two purpose, we started fabricating this device, and this device we called as a microelectrode array. Why microelectrode array? Because there are electrode which are in micron dimensions and it is a high density, but to start or to reach to humans we have to start from animal study. So, we will focus in this few lectures on rodent models. Rodent, we are using rat. Okay. So, we will see how we have fabricated the device and how device can be implanted, or rather implantation, again I will leave it on Dr. Sabri's lecture, but how once the device is implanted, how we are acquiring the signal, and how can we create a seizure or a fit or epilepsy in rodent models, and how our device can be used to understand the efficacy of anti-epileptic drug. Okay. So, these are few of the things that we will be discussing. So, if you see the slide, this slide shows electrocorticography signals that are recorded to study electrical activities of brain from the cortical surface, right. I told you what is ECOG, but more than that, let us first see this particular figure or schematic, or no, it is not figure, it is actually figure of a commercial available platinum iridium electrodes, which you can see here, and the electrodes when they are implanted onto the brain of a patient. So, the clinically validated implanted electrode arrays, which is let us say this B, used for pre-surgical diagnosis, have electrodes with diameter of 4 millimeter. So, each electrode is 4 millimeter in diameter, and the distance between two electrode is 10 millimeter. So, as I told you earlier, this array offers a poor spatial resolution during the pre-surgical mapping of the brain surface, and thus there is a very clear gap that we can find a solution to fill that gap, and that is that there is a need for electrode array with a higher density of electrodes to provide better spatial resolution in mapping brain surfaces. The application or one of the application can be epilepsy. Okay. So, with that, these are the gaps and what are the research goals and how the objectives can be designed and what can be the novelty of the device that we will be fabricating. These are some of the things which can be also of importance for students who are pursuing their master's research or PhD studies, to see that once you understand the gap, how can you fill the gap and how to put your slides together to understand the novelty factor, to understand what can be objectives to reach to achieve the aim, and what can be the research goals. So, the gap as clearly can be seen from the first statement. A high density ECG electrode recording can help clinicians to localize the epileptic focus in brain, but this is not there. High density electrodes are not available. Second, medication cannot improve the refractory or intractable, like I said, epilepsy. This requires a surgery to eliminate the region in brain. So, localization with a high resolution is important, which is not available or which is not possible with the current electrodes. So, the research goal can be to record the baseline electric activities with induced epileptic form discharges using ECOG in animal model, and to understand how the spectrotemporal analysis to determine the changes in electrical activities in different neurological conditions and distribution of signal over the cortical surface. So, once you have the device implant onto the rat's brain, let us say these are the electrodes touching the rat's brain, then one is that you can obtain the ECOG data, but second is to understand that if there is an episode somewhere, let us say this is the episode and this electrodes, let us say this one is 1, 2, 3, we can have many, but I am just giving some example, and this is not how the electrodes are actually so uniformly placed, because brain is extremely plastic, but let us say 4 and 5 is showing high electrical activity, right. So, you will see that other channels will show less and this will show very high activity like seizures, alright, like this, something like this, and we will see in the next slide. So, one is that to understand the high electrical activity, and second is to understand the effect of the existing drug. So, for this the objectives are to design and fabricate a microelectrode array, which is high density microelectrode array. In this case we say 32 channel is a high density, but you say, oh, 32 channel how can be high density, because we are talking about rat's brain, and for rat's brain is extremely tiny. So, for rat's brain 32 channel is a high density ECOG microelectrode array. And we have to implant this in rat's brain and we have to record and analyze the ECOG signals in response to the convalescent and anti-epileptic drug. What is convalescent? I am really sorry. So, the convalescents are the drug which will cause the epilepsy. Okay. So, bicaculin is one of the convalescents, and we can also give electrical stimulation to the rat's paws to create the epilepsy episode or seizures, okay, which is as per the literature. We do not do any novel drug formulations, but we rather focus on the fabrication of the device. So, once we implant the array in the animal model, what is the next thing? Recording and analysis of ECOG signal response to the convalescence and anti-epileptic drugs for detecting the epileptic focus and for studying the effect of anti-epileptic drug or efficacy of the AEDs, anti-epileptic drugs. So, there are multiple novelties, but let us understand first what are we talking about. What we are talking about is we will have a rat, we will do the craniotomy, implant the device. Once the device is implanted, we acquire the signal. You can see the baseline signal which you can see here, because these signals are acquired and processed through the Cyton Daisy biosensing board. Generally, Cyton Daisy biosensing board is used to acquire the EEG data from humans, but we have used this Cyton Daisy board to acquire the ECOG signal from rat's brain. Once it is acquired, it can be transmitted via Bluetooth using the USB dongle to the display, and we can see the ECOG data, right, channel wise on this display. Now, when we load the convalescents, or we can apply electrical stimulation to the rat's paw, the seizures would occur. That also we can see with the same device, which is implanted on rat's brain, and when we administer a drug which is anti-epileptic drug, the baseline is recovered. So, the 1 and 3 should be very close, the 2 are the channel, that is the first experiment is to acquire the signal which we call a baseline. Second experiment is to create epilepsy. Third one is to recover the baseline, not to actually recover, but to see whether the baseline is recovered or not. How? By using the anti-epileptic drug. So, a drug is effective, you will see that 1 and 3 would be kind of similar. Drug is not effective, then the seizures will continue, the epilepsy is not kind of treated. So, now let us understand the fabrication, okay, and you use all the understanding till now to focus on this slide. So, what we have done is you start with a silicon wafer, this is a base wafer and or a substrate, base substrate to hold the actual substrate which is our polyamide, but you have silicon. On this we can spin coat the polyamide which is here, and then we cure the polyamide, on which we deposit titanium and gold, because titanium is helps in improving the adhesion to gold. So, we have titanium gold, then this titanium gold everywhere is patterned, okay, is patterned, and then finally it is the electrodes. The electrodes are covered by another PI layer, and only the contact pads and the recording electrodes are opened, followed by realizing the device. So, now what I said, you see, once you pattern this, okay. So, this much is easy. What let us understand how to pattern it, okay. I will just give an example with one electrode and then we can take it further. So, what I said, we have a silicon substrate, on silicon substrate we have polyamide layer, okay, polyamide. On polyamide we are depositing titanium gold, all right, and then on this we spin coat photoresist, let us say it is positive photoresist, and on this photoresist we have the mask. Okay. This is a cross section, we have the mask. Now, of course, when we spin coat photoresist, what is the step? Step is that we have to soft bake, and soft bake is done at 90 degree centigrade, 1 minute on a hot plate. Now, this is our bright field mask, field is bright, pattern is dark, bright field mask. So, after soft bake we load the mask and then expose it with UV light. Then you unload the mask, develop the photoresist. So, we will unload the mask, develop the photoresist, develop the photoresist. What we will have? We will have this pattern, right, because it is a bright field. it is a bright field mask, and it is a positive photoresist. So, we will have photoresist like this, and then what we will do? We will do the hard bake at 120 degree centigrade 1 minute on hot plate, followed by, see, TiAu. So, Ti is at the bottom, Au is in the top, right. First is titanium followed by gold. So, first is Au etching. So, dip this wafer in Au etchant, then Di rinse, deionized water rinse, then titanium etchant, and again Di rinse, followed by drying in N2 nitrogen. If you do these steps, right, what we will have? The titanium and gold will be etched from the area which is not protected by the photoresist. Titanium and gold will get etched from the area which is not protected by the photoresist. Correct? We have seen this. Forgot the color. Okay, it does not matter. We will use the same color. Okay. So, the area which is protected by photoresist, you see the gold and titanium protected by photoresist is intact. The area which was not protected by photoresist got etched. How? First is gold etchant, gold is at top, followed by Di rinse, followed by Ti etchant, followed by Di rinse. Once you do that, then you dip this wafer in acetone. If you dip this wafer in acetone, what will happen? You know, acetone is a etchant for, or is a stripper for the photoresist. So, if a stripper for the photoresist will be stripped off. When photoresist gets stripped off, you have the following pattern which you can see here. This is your Pi and this is your TiAu, correct. Now, this figure, on this what we are doing, we are kind of covering silicon the Pi again, photoresist again everywhere, okay. Everywhere photoresist is deposited, so not photoresist, I am sorry, Pi, polyamide, because polyamide can also work as an insulator. And now what we are doing, we are opening the only contact pads, remaining area we want to have the polyamide. Why? Because you see here that this lines, right, everywhere the lines should be protected by the photoresist by the Pi, which is the insulating material, so that there is no short circuit. Only the contact pads, which is this one black one is the contact pads, and the recording electrodes 1, 2, 3, 4 all the way to 32. Only those electrodes, the Pi, which is the insulator, should be etched. Do you understand? So, first everywhere we have gold titanium pattern, TiAu pattern, followed by silicon dioxide everywhere. I am sorry, it is not silicon dioxide, insulator. Insulator is your polyamide. SiO2 is also insulator, but in this case we have used Pi. Okay. So, this is a insulator. So, first you deposit gold, pattern the gold, then spin coat the insulating material, which is the same material which is your base material, which is a polyamide. Okay. So, you spin coat polyamide, and then from the recording area, which is this area, the circles, okay, the circles are recording area, dots, and from the contact pads, because if you have insulator on the contact pad, you cannot take the contact. If you have insulator on the recording area, you cannot record. That is why the Pi, which is the insulator on the recording area and on the contact pad, we are etching. That is what it is here. Now you have Pi, and now you have to create a window, so that you can etch the PI from the substrate. So, let me now draw with so what we had, I will just put different patterns, so it is easier 3, 4 and 5 and then on that we had a PI and here also we have a PI, okay. PI on silicon, then PI as an insulator and here we have titanium gold electrode array. What we will do is we will spin coat photoresist, soft bake and load the mask, okay, and this will be our dark field mask, and then after loading the dark field mask we will expose the photoresist through the mask with the help of UV light, because it is our UV lithography, is not it. Okay. This is your dark field mask. If my and then what we'll do, of course we are forgetting one thing, okay. We are forgetting is there is a photoresist, because we have to spin coat the photoresist. So, this dot dot that I am drawing here is my photoresist, positive. So, on the PI, which is my insulator, we are spin coating positive photoresist, soft bake and followed by the UV light. Now, you know that the unexposed region will be stronger and the exposed region would be weaker, because there is a positive photoresist. So, we have a PI layer, we have gold titanium and then we had a second PI layer, right. So, and then we had a insulating we had the photoresist. So, this is a photoresist layer, because the unexposed region becomes stronger, right, and this cross that I am drawing here is a PI, right, because PI is an insulator, PI is the substrate, this is silicon, this is titanium gold. Now, what we will do? We will do the hard bake and then we will etch the PI, which is the etch the PI with a dry etching process. Okay. So, when you etch the PI with dry etching process, what you will have? You have PI, you have your titanium gold and you have your PI protected by the photoresist here, and from this area which is the area not protected by the photoresist, the PI is etched. right. The PI is etched. Now, we will dip this wafer in acetone. When we dip this wafer in acetone, what we get? We get our silicon, we have the PI, then we have our microelectrode array, recording electrodes and contact pads. Where there is no PI, right. So, here these are your microelectrode array and a recording, like contact and recording. So, this recording, forget about and recording and contact pads. Okay, recording and contact pads, and you can see that on recording and contact pad there is no PI, because PI is etched, right. So, after this we will we will etch the PI from that area which is not protected by the photoresist, you can see here, and followed by acetone dip. So, photoresist is stripped off and we have this particular thing where we have a microelectrode array, right, on PI, and the remaining area the PI is protecting the pattern. Okay. Easy. So, now if you see this particular figure again, you will understand it. I hope that you understand it little bit better, that you start with your silicon substrate, then spin you deposit your titanium gold, then pattern your titanium gold, then you deposit insulating material, and then you open the contact pads which is here, and or open the contact pads and recording area. Contact pads means, or electrodes means, that you remove the PI from the recording electrodes and contact pads, because PI is an insulator, followed by realizing the device by stripping the PI from the substrate, which is your silicon substrate. And once you have that, you will see you will get a flexible microelectrode array with electrodes and the contact pads, no insulating material. Everywhere else there is an insulating material. Now, the question is, why to have insulating materials everywhere else? Because when you implant, now in this case only this section is implanted, okay? Only this much section is, it goes into the brain, the remaining section stays out, because we are using 3.6 mm by 2.4 mm, which is slightly smaller than the rat's brain. So, it very well fits with the rat's brain. The point is, everywhere there is an insulator, only the recording electrode the insulator is not there, and the contact pad insulator is not there, because if the recording electrode insulator is not there, and then it can record well, it can acquire the signal, and there will be no short circuit between the electrode lines, because PI is there. What are other insulators? Silicon dioxide. What are other insulators? Silicon nitride. But in this case, a PI can act as a substrate and PI can act as a insulating material. So, this is the implantable microelectrode arrays, which is shown here in this photograph. These are the SEM image, scanning electron microscope image, and each electrode also you can see close to 50 micron in dimensions. Okay. So, we will stop here, and then in the next lecture we will see how to integrate this. Now, we know how the device is fabricated, right. How to integrate to a EIB board, right, into electronics interfacing board, and then how can we create a casing. So, the device can be placed through the casing that sits on the head, and the device should go on the brain, right, and then how can we connect it with the open cyton BCI system. So, the idea is to not just show you the device, but also to take further at how to integrate device and acquire the signals, and then perform the experiments and understand how the device is performing. So, we also have recordings for wherever possible for the surgery. We have recordings for the all the PVD, we have recordings for photolithography, right. We have recordings for 3D printing, so that it will give a understanding about different tools that needs to be used for fabricating and implanting these devices or microelectrode arrays into the brain, in this case of a rat. So, we say rodent models. So, with this I will leave you here, and I will see you in next lecture with more discussion about neural implants. Till then, take care. Thank you very much.